The overall goal of the following experiment is to examine the neural substrates of aware and unaware fear learning and memory by simultaneously measuring skin conductance response, conscious unconditioned stimulus, expectancy, and functional MRI. This is achieved by measuring skin conductance response, or SCR, as an index of fear learning and memory. Unconditioned stimulus expectancy is also monitored, which reflects the participant's expectation of receiving the aversive unconditioned stimulus and can be used as an indicator of the participant's awareness of stimulus contingencies. Further, fMRI data are acquired in order to identify learning-related differences in brain activity when subjects are aware compared to unaware of the relationship between stimuli. Results typically show greater hippocampal responses to the positive conditioning stimulus, or CS+, than to the negative conditioning stimulus, or CS-, on perceived but not unperceived trials. However, amygdala responses are larger to the CS+ than CS- on both perceived and unperceived trials.
The main advantage of this technique over other methods of assessing awareness, such as post-experimental questionnaires, is that awareness can be assessed on a trial-by-trial basis during the conditioning session.
This method can help us answer key questions related to learning, memory, and emotion, such as what role contingency awareness plays in fear conditioning.
Physiological monitoring systems are non-standard equipment in most imaging facilities. Therefore, schedule 15 to 30 minutes prior to participant arrival to set up